Hello students, now we will start with new topic that is about slime molds. We already have seen that slime molds are a part of protista, means they are studied under protista. Under protista, what do we read? We read the chrysophytes, dinoflagellates, euglenoids, and also slime molds. Next one going to be read is protozoans. So now we will start with slime molds. Now, what is habitat of slime molds? Slime molds are saprophytes that live in dead and decayed matter. These slime molds, nothing but they are equal to that of fungus. See, in the fields or in some area where there is litter which is prone to moist in condition and it is getting decayed, in that area these slime molds will grow. So they depend on decomposing litter. Litter means the fallen leaves and the waste parts, twigs, etc. from the plants. Decomposing litter and decaying organic matter. So here we have used a term like these are saprophytes. It means that the organisms which depend on decaying and dead organic matter and then converting into useful minerals. Such types of organisms are called as saprophytes. We use this saprophytes term to fungus. So their adult and reproductive body is called as plasmodium. So plasmodium usually we read till now under protozoans, but here the adult body of slime molds is also called as plasmodium. Now plasmodium is an aggregation of slime mold formed under favorable conditions. This plasmodium, its nutrition is by absorbing of the organic matter from the decaying matter. Plasmodium of slime molds does not have true cell wall. Roughly, we can give a diagrammatic representation like this will be the structures, root-like structures which are fixed into the litter. We can say these are a type of hyphae endings. Now this will be the plasmodium. It will not be having any specific shape. It will be zigzag in manner. It will move towards the direction where food material is available. We can also use a term like this is a matured plasmodium. During unfavorable conditions, this plasmodium differentiates and forms fruiting bodies. Which bears spores. Now, if we say that this is during favorable condition, but when conditions are not favorable, then the same plasmodium will be converted into other different fruiting body form, means it is ready for reproduction. So it produces special types of structures like this, and these structures are called as spores. So these are spores. Now, during favorable conditions, it is normal plasmodium. But now when it is bearing the spores, it is called as fruiting body. Nothing but the plasmodium, but now it is in a stage of production of spores. This is the active form or fruiting body. Now, these spores are at tip of the fruiting body. These spores have true cell wall. Plasmodium in general, an adult plasmodium of the slime molds will not have true cell wall, but the spores that are formed during unfavorable conditions are having true cell wall. This is important point to remember. So what is use of the cell wall? Cell wall helps the spores to overcome the adverse conditions or the abnormal situations. That is use of cell wall on the spores. So matured plasmodium, which is having roughly a structure, which these are the root-like structures that are formed into firmly fixed into the substratum. Now, during unfavorable conditions, now the same matured plasmodium, which is in favorable conditions, the same one, it will now be called as a fruiting body. It bears the spores at the tip of the fruiting bodies. So now these are spores. Now these spores, when germinated, they give rise to two different types of cells. What are those? One is amoeboid form, another one is flagellated form. So this is amoeboid form, this is flagellated form. So this will be nucleus. So again, many number will be formed like this, many flagellated form, many amoeboid form. They both tend to fuse with each other to form new colony of the slime mold. They both fuse and get fertilized to form zygote. So now this is haploid in condition, this is haploid in condition, spores are haploid in condition, fruiting body diploid in condition, plasmodium diploid in condition. When these both are fused, we get diploid condition zygote. Now this zygote will be again developed into plasmodium. Develops into plasmodium. So this is a brief explanation of how slime molds are developing their number. So here we can see how many stages of haploid condition stages are there. Spores 1, amoeboid 2, only 2 stages. So here we can say up to here. Here we can say this is N condition and other one is diploid condition.